Historically, in light microscopy, we're interested in how the path of light changes through a sample. This can be through reflection or absorption, or even through bending of the light, like you would see through a prism or a glass of water. And what we see at the end of this is a pattern of light and shadow. But if you're familiar with these beautiful images of our microscopic world, they're usually full of color. How do we accomplish this? Well, like most of our ingenious ideas, we stole this one from nature. There are many organisms or animals that can make themselves glow, like glowing jellyfish in the deep sea or fireflies on land. And this glow is called fluorescence. What's happening is the animal is able to absorb energy of one form and then use that in a material within their bodies to emit light of a specific color. And now we can make materials within cells and tissues under a microscope do the same thing. So how do we accomplish this? The spectrum of visible light goes from blue, which is relatively higher energy, to red, which is comparatively lower. When a fluorescent material absorbs that higher energy light, it only holds on to it for a second and then emits a lower energy light, or a color shift, plus a little bit of heat. So now, instead of watching light get reflected or bent, we're really looking at only those wavelengths that are emitted from the fluorescent material. A simple at-home example of this is actually tonic water. If you shine an ultraviolet light on a glass of tonic water, the molecules in there, called quinine, can absorb that ultraviolet light, which is higher energy, and emit blue light, which is a little lower, but it's within the visible spectrum, so suddenly we can see it shine. Decades of research have led to the modification or even invention of new fluorescent materials that can brilliantly shine in a whole rainbow of different colors. The wonderful thing about this is it means we can take these different materials and put them into our samples at specific locations. We'll know, for instance, that the DNA is going to shine blue, or that the structural components of the cell that hold it in its shape will shine red. We can visualize this using a fluorescent microscope. A fluorescent microscope has a light source that emits a single color of light onto the sample. The fluorescent material inside that sample will emit its own shifted color, which we then collect with a series of lenses and filters. What we're left with at the end, what hits the camera, is just a black background and just the emitted colors that we were expecting to see from our fluorescent materials. While these images are certainly beautiful to look at, they're also supremely informative. We can watch cells pulling together to close a wound, or many other aspects of this dynamic process we call life. Modern biology wouldn't be where it is today without being able to use fluorescence to make our invisible world stunningly bright.